Reproduction and dissemination of pathogen. Introduction. Plant pathogens, which include bacteria, fungus, virus, nematodes, insects and pests, etc., play a major role in disseminating plant disease. Each of the above has a unique mode of reproduction and specialized structures to ensure that the disease-causing agents is transmitted to other plants. Natural agents like water, wind, soil also play a role in the same. With advent of technology and transport systems, man himself contributes in a major way by transmitting diseases across oceans and mountains. Reproduction of bacteria. In general, bacteria reproduce by binary fission, one cell splitting into two. Many plant pathogens harbor plasmids, example strains of Pantoia, Stewarti subspecies. Horizontal transfer, the passage of DNA from one bacterial cell to another, may be accomplished in the laboratory and is assumed to account for much of the genetic variation among strains of a species and even among species in nature. Bacteria may also harbor prophage, inherited form of a bacterial virus or remnants of prophage integrated into their chromosomes. If a whole phage is integrated, some cells may lyse or break open naturally or can be induced to lyse by chemical treatment. Example, mitomycin C. In a very rare case, apparently both pathogenicity factors and a potent mammalian toxin are carried by a non-integrated bacterial virus in Rathibacter toxicus. Fungal reproduction. Fungi frequently reproduce by the formation of spores. A spore is a survival or a dispersal unit consisting of one or a few cells that is capable of germinating to produce a new hypha. Unlike plant seeds, fungal spores lack an embryo, but they contain food reserves needed for germination. Many fungi produce more than one type of spore as part of their life cycles. Fungal spores may be formed via an asexual process involving only mitosis, that is mitospores, or via a sexual process involving meiosis, which is the meiospores. Many fungi produce spores inside or upon a fruiting body. Mushroom is a type of fruiting body produced by some Basidiomycota. Other fungal fruiting bodies are puffballs or shelf fungi. These are examples of large conspicuous fruiting bodies, but there is an even greater diversity of microscopic fruiting bodies produced by various fungi. One thing which all fruiting bodies have in common is that they produce spores and provide a mechanism for dispersing those spores. Survival and dissemination of virus. Plant viruses are obligate biotrophic parasites. Their life cycle starts by penetration of the virion into the cell. Plant viruses are unable to penetrate the plant cuticle and cell wall. It is believed that the virion enters the cytoplasm of the cell passively through wounds caused by mechanical damage to the cuticle and cell wall. The next phase of virus infection is the partial or complete removal of the coat protein shell of virion in the cytoplasm. Next, the cell mediates expression of the viral genome by providing a transcription apparatus for DNA viruses and a translation apparatus for all viruses. The DNA viruses must be transported to the nucleus for transcription in order to gain access to the cell proteins required for the production of messenger RNA from viral DNA. Translation of viral RNA in the cytoplasm produces viral proteins that are required for completion of the virus life cycle. Only few viruses such as TMV rely on long term, which can be up to many years of survival in the environment, and on passive mechanical transmission from plant to plant. Most plant viruses are actively transmitted from infected to healthy plants by a living organism called a vector. Plant feeding arthropods, nematodes, and plant parasitic fungi are major types of vector organisms for plant viruses. Among this, aphids and whiteflies are capable of transmitting the largest number of virus species. Most viruses are actively transmitted to healthy plants in a matter of seconds, hours, or days by vectors. In addition, some viruses are transmitted from infected plants by vegetative propagation, for example, through tubers or grafting, or through seeds or pollen from infected plants. This type of transmission is extremely important for virus survival during the winter. Transmission of viruses by a vector is very specific process. Each particular virus can be transmitted by only one vector type. Example, aphid, and not by another, which could be a whitefly. Conversely, each vector species, example, green peach aphid Mysis persicae, can transmit some viruses, which is the beet yellows virus, but not others. The citrus tristeza virus, despite the fact that these viruses are genetically similar to each other. The interaction between a virus and its specific vector that results in virus transmission varies for different virus vectors. The relationships that have developed between viruses and their vectors are complex and of considerable interest because vectors provide the main method of spread of disease for many viruses that cause severe economic losses. For some virus vector combinations, the interaction between a virus and its vector is very superficial and is the result of attachment to the external surfaces of the vector mouthparts. Viruses in the potyvirus genus produce a special protein called the helper component that glues the virions to the aphid stylets. In this case, acquisition of the virus from infected plants and inoculation of virus to the healthy plants takes from seconds to minutes. In contrast, viruses in the family Luteoviridae circulate in their aphid vector, moving through the gut wall into the body cavity and then into the saliva of the aphid. It takes around 12 hours for these viruses to circulate in their aphid vector before they can be transmitted to another plant. In other plant virus vectors, the viruses actually multiply in the cells of the insect vectors. For example, tomato spotted wilt virus multiplies in cells of its Thrips vector, and once Thrips acquires this virus, they transmit it for the rest of their life. These plant viruses that multiply in their insect vectors are considered to be both plant and insect viruses. Transmission of plant diseases. Rational and most effective control of parasitic plant diseases is possibly only if the disease is correctly diagnosed, the nature of transmission of the disease is known, and the life cycle stages of the involved parasite, its mode of reproduction, and active structures produced under the favorable condition for wide dispersal and the structures produced to overcome adverse condition are known well. All parasitic as well as viral diseases are they are primary infection. Contact of a pathogen with a suitable host plant and the initiation of a disease first time in the season of a crop is called a primary infection. Often a few or several plants in the crop are likely to get primarily infected. Secondary spread. When a plant or few plants are primarily infected, rapid multiplication of the pathogen sets in under favorable climatic conditions which help the secondary spread of the disease. In case of fungal diseases, the primary infections are usually caused by the resting or dormant structures of the fungal pathogen, while the secondary spread occurs throughout the asexually produced countless and short-lived spores. Although in majority of the plant diseases, the above two phases occur sequentially, primary infections occurring only once in the season and secondary spread often occurs several times in the same season, thus causing rapid transmission over wide areas. Mildews, leaf spots, blasts, blights, etc. are the common examples of former type, while some smut disease of cereal or grain crops where black powder is formed in place of grains fall in the later type. Control measures therefore in the former type need to be directed to avoid primary infection and also to check the secondary spread. Let us now look at the modes of primary infections. According to the mode of primary infection, plant diseases can be classified as soil-borne, seed-borne, including diseases carried with planting material, wind-borne, insect-borne, etc. This knowledge is essential in adopting suitable control measures. Continuous and discontinuous transmission. Transmission of disease is said to be continuous when it occurs naturally by way of growth, multiplication and spread of the pathogen in an area or country where the disease is established. At times, however, in an area or country where a particular disease has never occurred, it may get introduced through the agency of man carrying disease material to a new locality or to a distant country for the purpose of introduction of new plant crops, varieties, etc. Such transmission is unnatural and is said to be a discontinuous transmission. Quarantine regulations are enforced to check such discontinuous transmissions. Direct and indirect transmission. Disease transmission can also be classified as direct transmission. Disease transmission, where the pathogen is carried externally or internally on the seed or planting material like cuttings, sets, tubers, bulbs, etc. Indirect transmission. The pathogen which spreads by itself by a way of its persistent growth or when certain structures of the pathogen are carried independently by natural agencies like wind, water, animals, insects, mites, nematodes, birds, etc. are said to be the different methods of indirect transmission. Direct transmission. Internal transmission through seed. False smut disease and helminthosporin blight disease of wheat are the common examples of fungal diseases carried internally through apparently healthy seed. Ring rot and brown rot of potato caused by bacteria are carried internally through the tubers. The well-known whip smut and red rot of sugarcane are fungal diseases carried internally in the planting sets. Mosaic and leaf roll of potato, which are viral diseases, are also carried inside the infected tubers. External transmission through the seed. In this mode of transmission, the pathogen is carried externally over the surface of the seed or through vegetative propagation through tubers, bulbs, etc. They may even be carried as a physical mixture of fungal structures with the seed. The common grain smut of jowar is an example of the former type, while the fungal structures called sclerotia have the size of a grain or slightly bigger in case of the ergot disease of bajra are often likely to be transmitted in the form of physical mixture within the seed. Indirect transmission. Indirect transmission can also be termed as autonomous transmission. It takes place by continuous and persistent growth of the threads or hyphae of the causal fungi in soil, characteristic of several wood-rotting fungi attacking forest trees and some fruit plants. Some root-rotting fungi infecting certain seasonal crops are also transmitted by this method. The autonomous dispersal of such soil fungi may range from a few centimeters to about 8 to 10 meters in a single season. Some plant parasitic nematodes also exhibit active but limited mobility in the soil. Wind dispersal. This is a common mode of transmission. Fungal spores produced externally on host surfaces are easily carried by wind currents, and this is the most dangerous mode of transmission of plant pathogenic fungi like those causing powdery and downy mildews, leaf spots, blasts, blights and rust diseases. The black stem rust disease of wheat in India, on wild grasses in the Nilgiri hills in the South India, from where the rust spores are carried to South, Central and then to North India by wind currents every year. Spores may be carried from low to very high altitudes of 12,000 to 14,000 feet and from short distances to very long distances of several hundred kilometers. Extensive and severe epidemics of plant diseases are mostly the results of wind transmission of the pathogens. Wind dissemination involves 4 stages relating to the spores. They are production of the numerous spores, their liberation in the wind currents, dispersal and deposition on the new susceptible host surfaces where they can cause infection under favorable climatic conditions. Apart from spores, bits of fungal threads and nematode cysts are also exposed to wind transmission in certain cases. Water dissemination. Disease transmission through water in different ways is comparatively less as compared to the wind transmission. Splashing raindrops mostly transmit the foliar diseases from leaf to leaf, from shoot to shoot and even from plant to plant in case of closely spaced crops. Such transmission is usually accompanied by wind dispersal as well. Plant pathogens requiring high humidity conditions like the fungi causing downy or mildew disease or bacteria causing canker of citrus are well adapted to this kind of short distance water dispersal. Certain soil inhabiting pathogenic fungi and bacteria causing collar rots, wills, foot rots, etc. are likely to be transmitted to much longer distances through irrigation water, streams and rivers. Water dissemination plays an important agency in transmission of seeds of higher flowering parasites like the dodder. Animals. Farm animals serve as disease transmitting agents in many cases. They play a role by carrying the pathogen externally on their body surface, particularly on legs and hooves, or internally through their intestinal tract. Commonly, the soil inhabiting fungi causing rots and wills are carried externally, while certain smut fungi causing diseases to grain crops are transmitted to the intestinal tract. Birds. Although birds play a very minor role in disease transmission, in cases of dispersal of seeds of higher flowering parasite, Loranthus species parasitizing certain trees like mango, their role is of great significance. They transmit Loranthus both externally and internally. Farm equipments and tools. Farm equipments used for cultivation of soil are often involved in transmitting plant pathogens from one place to another. The pathogens in this case are usually carried in the form of bits of plant disease debris lying in the soil. Similarly, tools used for carrying out operations like cutting, pruning, budding, grafting, thinning also help in transmission of certain diseases from plant to plant. Several viral diseases are disseminated through the budding and grafting operations. Insects. Most of the viral diseases of plants are transferred to the species of aphids, jassets or leaf hoppers, white flies, mealy bugs, etc. Certain bacterial and several fungal pathogens are also known to be carried by insects. Mites. Mites are wingless arthropods resembling ticks and having 4 pairs of legs and no antennae. It is suspected that some viral diseases of chilies, tomato, brinjal, etc. are caused by mites as vectors. Nematodes. Nematodes have been observed to transmit viral, bacterial and fungal plant diseases. Nematodes which feed externally on host plant roots cause injuries to roots which become the avenues for entrance of fungal and bacterial pathogens and hence causes infection to the roots of plants. The fan leaf virus of grapevine is a well-known example of transmission through a species of nematodes. Biological transmission. Dodder, which is a higher flowering parasite, is known to transmit certain viral diseases which remain persistent in the dodder plant. The flowering parasite, after acquiring virus from infected plant, does not show any symptom by itself but remains capable of transmitting the virus to healthy hosts. Human dispersal. Man is often responsible for transmission of plant diseases mainly in 2 ways. Workers handling seeds, other planting material or fruits come in contact with plant pathogens like fungi or bacteria. While handling the diseased material, most of the times unknowingly and indirectly transmit the pathogens to healthy seedlings or plant parts through his contaminated hands. This is again a continuous mode of transmission. The other or discontinuous mode of transmission for which man is responsible is the most efficient and equally dangerous phenomenon of transmission of plant diseases between distant geographical areas often separated by physical barriers like oceans, mountains or deserts. Such long distance transmission of a disease to an area or country which is free from the disease usually occurs by the transport of infected seed, nursery stock or timber. In the history of plant pathology, this mode of transmission has often resulted into some of the worst plant disease epidemics in new areas. The local host plant stock being not adapted to the pathogen and consequently remaining highly susceptible. Fungal diseases like late blight of potato and downy mildew of grapes, bunchy top of banana, which is also a viral disease, and bacterial blight of paddy are examples of some severe and important diseases which were introduced in India from other countries. Many countries of the world have suffered from introduction of such species through man. Therefore, many international authorities have taken initiatives in promoting studies in this regard and designing regulations to reduce the danger.